It's my great pleasure to welcome all of you to the Sam Eiduson Lectureship. The Sam Eiduson Lectureship, as you sort of can guess, is named after Sam Eiduson, who was born 100 years ago, but only passed away 11 years ago. So he lived a very long life, and he spent most of his life actually here at UCLA, working in different capacities. He was the first person to hold the education associate chair in neuroscience in education at the Brain Research Institute. When he was doing that, he started sort of his own field. He's the one that generated, or is at least one of the ones that generated the field of neurobiochemistry. So he was a professor in chemistry, in biochemistry, and then got associated with the Brain Research Institute. And as such, felt that really what was necessary was to have a PhD track in neuroscience. And he was one of the founding members of the Neuroscience interdepartmental graduate programs here at UCLA. So that started in 1972, depending on who you read online. It could have started in 1968. So around there is when the Neuroscience interdepartmental program started. The Sam Eiduson Lectureship started in 1993, and now this is the 26th lectureship in his memory. As I alluded to, he passed away in 1907, but while he still was capable, he actually came to each and every one of those lectures, and he sat very much in the front and was active, engaged, and really participated in listening to the neuroscience. The lectureship is given by a committee. I'm not part of the committee, so there are others of you who really are responsible for that. It's given to what we consider the best thesis or contribution within neuroscience that particular year. It's not limited to students in the Neuroscience interdepartmental program, so anybody of you amongst the students who feel they're really contributing to neuroscience, as long as your PI is in the Brain Research Institute, you're eligible to win this prize. This year, it's my great pleasure and honor to actually welcome somebody of outside of the Neuroscience interdepartmental program, Celine Vuong, who has done some absolutely fantastic work that we'll hear about in Doug Black's lab. And I let it be to Doug to actually introduce Celine a little bit more closely. But before we do that, I can give to Celine the really wonderful plaque that she can mount in her postdoctoral lab. She's going just a little bit south of here after some excursions that she might be able to tell you outside. So after this lectureship, we actually have a little reception outside with food and everything. So congratulations to the 2018 Sam Eiduson Award.
Thank you.
And with that, we give it over to Doug Black, who will introduce Celine, and I will not introduce Doug.
That sounds good. Yeah, so I'll make this quick. So thank you to Felix and the Eiduson Committee. This is really for honoring Celine in this way. It's really exciting for me. I think as a biochemist who wandered into the world of neuroscience, I think that it's sort of fitting, myself being the biochemist. Celine, she is from San Diego. She went to Brown University, and then she went and was a tech and lab manager for a couple of years in Azad Bani's lab at Harvard Medical School. And there she learned a lot of neuroscience and worked on cerebellar development before she came here to UCLA to go to graduate school. And she came to UCLA really saying she was interested in two things that still seem to hold true, neuroscience and post-transcriptional gene regulations, or gene regulation at the RNA level. And so when she came into the lab, she was really attracted to questions related to physiology. And I've always tended to shy away from some of those questions in our systems, that the RNA-binding proteins we study have lots of targets, and it seemed like it would be very difficult, to me it seemed very, very difficult to try to pick apart the phenotype of an RNA-binding protein mutant in relation to a particular target and its effect on physiology. But in fact, as you'll see, this is exactly what Celine did. And she did this really, I think, by making a number of really key decisions in her project about how to move it forward. And so she set up a number of critical collaborations with members of our neuroscience community that gave us a lot of help on the physiology and other techniques. She really has remarkable technical skills and ability to make really hard experiments work that I think you'll see. And then more recently, she's really showed a lot of patience in the review process, and that when the reviews come back that ask for the moon, she really didn't complain very much. She just went and got the moon and gave it to them. So as you'll see, she did some pretty hard things. And so I think Celine is one of these students that I can say has probably taught me as much as I've taught her. She really has a great understanding of her project in neuroscience at large. And so this talk is really a nice going away for Celine. She's going to start her postdoc next month at the Salk, and happily that's not too far away. So I have some sense that she might come back to visit us frequently. So and Celine, welcome. Thank you.
So thank you, Felix and Doug, for those very kind introductions, and thank you to the award committee for giving me this. I'm very happy that I can share my graduate work with you all today. All right, so today I'll be talking to you about my graduate work titled Rbfoxl regulates inhibitory synaptic transmission through the v-SNARE Vamp1. So I don't think I have to tell this crowd that the brain is an amazing organ, which allows us to process sensory information as well as think of very large ideas. But one of the big questions in neuroscience, of course, is how do we go from a bunch of cells to something that allows us to do these very complex processes and calculations. So I'm sure you all know that the units of the brain are the neurons. And each neuron receives information from another neuron at synapses, which are located either on dendrites usually or on the cell body. And then after receiving this input, neurons collate all that information together and then decide whether or not to give an output in the form of an action potential, which travels along the axon and ends at the axon terminal with release of neurotransmitter to another receiving neuron. So one broad category of the many types of neurons that are present in the brain are excitatory and inhibitory neurons, which generally give off different neurotransmitters, which in turn either promote or reduce the likelihood of the receiving neuron producing an action potential. And so the way that these neurons are arranged in a circuit and the strength and the speed of their connections are really what give rise to the brain's ability to process information and output information. So, of course, dysfunctions of neural circuitry can lead to very serious issues. Among them are the autism spectrum disorders, which are gaining traction. About 1 in 68 children in the US are affected by this. Another large disorder affecting many people is epilepsy, which affects about 50 million people worldwide, and this is data from the NIH and the World Health Organization. So historically, the focus on understanding how these serious neurological diseases arise have focused usually on one single genetic cause, but recently there's an increasing recognition that there are multiple factors at play in these very complex diseases, And one factor that is of increasing interest is the contribution of RNA metabolism and therefore RNA-binding proteins in the cause of these complex diseases. So let's just backtrack a moment to the central tenet of molecular biology, the central dogma, which tells us that DNA, or our genes, is transcribed into RNA, and that RNA is then translated into proteins, which do the bulk of the work in the cell to perform maintenance and also activity-regulated functions. While this dogma seems simple, if you just focus on the RNA step, it in fact consists of a myriad of many, many steps that affect every part of the RNA's life from the point of its conception at transcription all the way to the end at degradation and turnover. And the proper regulation of RNA metabolism is a crucial point for proper function of any cell. So it's no surprise then that RNA-binding protein dysfunction affects neurons very much. In the neuron especially, the proper function is very dependent on RNA-binding protein regulation. So this includes things such as regulated alternative splicing, polyadenylation, especially in neurons, the subcellular localization and transport of RNAs to particular sites and their local translation in response to activity, as well as regulated degradation and turnover. So the disruption of RNA-binding protein function and therefore RNA metabolism can lead to many neurological problems. As you can see that RNA-binding proteins are active in every step of neurogenesis, all the way to migration, synapse formation, and the mature function of neurons. So in the Black Lab, one of the families of RNA-binding proteins that we study is the Rbfox family. So there are three members of the Rbfox family in mammals, Rbfox 1, 2, and 3. They are highly enriched in neurons. They all contain a highly conserved RNA recognition motif, or RRM. And it's this RRM which binds to and recognizes the sequence UGC-AUG. So an important point to know about Rbfox is that my project focuses on Rbfox1 specifically, and Rbfox1 has two main isoforms. It has a nuclear isoform and a cytoplasmic isoform. So the nuclear isoform performs regulated alternative splicing, whereby binding downstream promotes the inclusion of an alt exon, and binding upstream of Rbfox1 suppresses the exon. While in the cytoplasm, Rbfox1 also acts at the 3' UTRs of its target transcripts to promote mRNA stability and increase gene expression. So previously in our lab, we had characterized an Rbfox1 conditional knockout mouse, and this mouse was crossed to a paneuronal nesting Cree. So the resulting knockout mice have spontaneous seizures and neuronal hyperexcitability. To study this susceptibility to seizure, we performed canic acid injections which induce seizures, and from this data we saw that the Rbfox1 knockouts, shown here in black, are susceptible to more severe and lethal seizures as compared to their wild-type littermates or their Rbfox2 knockout counterparts. Then to better understand the electrophysiology of how these seizures come about, in a collaboration with the Modi lab here, we performed recordings from the dentate gyrus, where the lateral perforant path in the hip is stimulated, and then field recordings are made in the dentate gyrus. And these experiments essentially told us that with any given stimulus, the Rbfoxl knockouts have a higher response, a larger response compared to their wild-type littermates, showing that in general, the Rbfoxl knockout neurons are hyperexcitable. So when I came to the lab, I was very interested in the Rbfoxl knockout, and the main question that I had was what are the molecular mechanisms that allow Rbfoxl to regulate neuronal excitability? And in the first part, I will focus on the mechanistic work that we did to understand this. And the two sub-questions we had were which Rbfoxl targets are contributing to this increased neuronal excitability in the knockout, and for these targets, how is Rbfoxl regulating them? Is it through alternative splicing, or is it through maintaining mRNA stability? So the previous work that I had just mentioned to you had identified some Rbfoxl splicing changes in the knockout using microarray. However, given recent work implicating cytoplasmic Rbfoxl function, we wanted to use a method where we could see both splicing and gene expression changes. And we wanted to focus on a particular portion of the brain, the hippocampus, where we had previously seen electrophysiological changes. So to do that, we dissected hippocampi from adult wild-type and Rbfoxl knockouts. We did basic poly A+ paired and 50 base pair sequencing. And from this data, we could do splicing analysis using the splice trap program, or we could do differential gene expression analysis using cufflinks. And we decided to focus on the differential gene expression changes, given the interesting recent work that was published. And we were also interested in knowing which of these changes were resulting from direct Rbfoxl targets. And to do that, we decided to combine the gene expression changes with Rbfoxl iClip in the 3' UTR. So for some of you who may not be familiar with this method, iClip is essentially a way to UV cross-link RNA-binding proteins to their RNA targets, IP it, and then do high-throughput sequencing to identify where in the genome the RNA-binding protein is binding. So we combined our gene expression changes with Rbfoxl binding data, and from that we could do a very, very stringent combination to find 15 high-confidence Rbfoxl targets that are changing in expression. And so those 15 targets are shown here. Most of them, the ones highlighted in yellow, are decreasing in expression with loss of Rbfoxl. And this is the general trend across all of our gene expression changes that we saw in the list that has less stringency. And we decided from this list to just focus on one particular target, Vamp1. And the reason is that it is, the top changer with all these requirements, but also, as you can see, as you'll see later, it has very interesting physiology that made a lot of sense in the context of the Rbfoxl knockout. Right. So from all of our RNA Rbfoxl iClip data and our gene expression data, we saw that Vamp1 was a direct Rbfoxl target. So here I'm showing you a genome browser view of all this data. So on the bottom here you have the gene structure of Vamp1. The boxes here are the exons, the dashed lines are your introns. This is your 3' UTR. Above that we have the RNA-seq data from the Rbfoxl knockout and the wild type. As you can see from before, with loss of Rbfoxl, there is a significant decrease in expression across Vamp1. And then here at the top, we have Rbfoxl binding data from the iClip experiments showing that Rbfoxl binding is found mostly in the Vamp1 3' UTR and close, some of them are close to UGC, AUG, the Rbfox binding motif, which is shown here in black. So we confirmed our sequencing results by doing qPCR in the hippocampus, the cortex, and the cerebellum of wild-type and Rbfoxl knockout mice. We basically saw the same as in our sequencing, where loss of Rbfoxl in the forebrain results in a significant decrease in Vamp1 mRNA. And this was preserved at the protein level, where again in the forebrain, loss of Rbfoxl leads to a significant reduction of Vamp1 protein. So one major question is, how might Rbfoxl be regulating the expression levels of Vamp1? So two recent papers gave us some idea of how this might be working. So in the case of neurons, cytoplasmic Rbfoxl has been shown to act on its target transcripts by binding to their 3' UTRs and promoting expression of those targets in part by blocking the repressive activity of the RISC complex and targeting microRNAs. However, Rbfoxl can also act in the reverse direction, blocking expression, and this was shown in the developing Drosophila embryo, where Rbfoxl activity at the target 3' UTRs actually blocked translation of those particular targets. And in both cases, this is mediated by the cytoplasmic isoform of Rbfoxl. So of course we wanted to know, how is Rbfoxl acting on our target, Vamp1? So to test whether or not cytoplasmic Rbfoxl was responsible for the regulation of Vamp1 expression, we took cultured hippocampal neurons from wild-type and Rbfoxl knockouts, and then we tested, again, Vamp1 mRNA levels by qPCR. We see a significant reduction with loss of Rbfoxl, as we saw in the brain. And we find that if we add back only the cytoplasmic isoform of Rbfoxl, we can rescue both the mRNA and the protein levels of Vamp1 back to levels similar to that of the wild type. So in this experiment, we show that cytoplasmic Rbfoxl is enough to promote the expression of Vamp1 at both the mRNA and the protein level. So then the next question was, which portion of the Vamp1 transcript is responsible for this regulation? And based on previous work, we had guessed that this would probably be the 3' UTR. So to test that, we set up this experiment, which is a luciferase to test the 3' UTR. We cultured floxed Rbfoxl hippocampal neurons, to which we infect them with either a control virus or a Cre virus to make wild type or Rbfoxl knockouts. And then to this, we transfect a luciferase reporter with the Vamp1 3' UTR attached. Right, so we first tested the full-length Vamp1 3' UTR, which contains 7 potential Rbfox binding sites. Similar to what we saw with our protein and mRNA analyses, the loss of Rbfoxl results in a dramatic decrease of reporter expression, presumably from loss of Rbfoxl binding. If we mutate the 6 upstream sites of this reporter, we find that there is a significant reduction in reporter expression, presumably because we are losing quite a lot of Rbfoxl binding. If we mutate all the sites, we completely lose any expression, showing further that you need Rbfox binding in general to promote expression. And finally, we were particularly interested in what happens when you mutate only the very 3' or last Rbfox site in this UTR. And we found that mutation of this last site actually quite dramatically reduces the reporter expression indicating to us that this last site plays a large portion in promoting expression through this 3' UTR. So why would this particular 3' site be so important? So when we looked closer at this area of the UTR, we found that this Rbfox binding site is right next to a miR9 binding site, and presumably binding of Rbfox1 here likely prevents binding of miR9 and the RISC complex at the same time. So to test this, we made a second reporter containing only this 3' segment, and we find that, again, similar to the full-length 3' UTR, loss of Rbfox1 results in a significant decrease in reporter expression, presumably because normally you would have Rbfox1 blocking miR9 repression. We find that if you mutate that same 3' Rbfox site, similar to the wild type, you get a significant reduction in expression because you can't have Rbfox1 binding there, blocking miR9. And finally, if you mutate the miR9 binding site, we can get a significant increase in expression, indicating that likely the competition between Rbfox1 and miR9 at this particular site is what is maintaining expression of the reporter. So finally, to just hammer this down even more, we wanted to know if blocking miR9 would increase expression even in the absence of Rbfox1. So to do that, we just use AAV expressing this tough decoy, which is a hairpin containing a binding site for mature miR9 to basically soak up the microRNA and reduce its levels in the neuron. And using this, we can significantly reduce miR9 levels to about half in our neurons. So using the same full-length reporters, we find that in combination with the tough decoy, we find that with loss of miR9 using this tough decoy, we can increase the expression of the reporter even in the absence of Rbfox1. And if we look particularly again at the reporter where you have the last Rbfox binding site mutated, blocking miR9 even in the absence of Rbfox1 can significantly increase the reporter expression. So together, these very long luciferase assays tell us that binding of Rbfox1 to the upstream binding sites in this 3' UTR work to promote expression, possibly by increasing translational efficiency, but a large portion of that regulation is likely due to the opposing effects of Rbfox1 and miR9 at the very 3' end of this 3' UTR. So just to summarize for you the first part, which is the answering how the mechanism of how Rbfox1 regulates Vamp1 expression. We identified multiple direct Rbfox1 targets in our pan-neuronal Rbfox1 knockout mouse. We found that Rbfox1 can promote Vamp1 expression, particularly through the Vamp1 3' UTR, and that a conserved 3' Rbfox binding site promotes expression in part by blocking the repressive action of miR9, and that blocking miR9 can restore some of the expression even in the absence of Rbfox1. All right, so the salient question after all these mechanistic studies, which was what electrophysiological functions are altered by loss of Rbfox1 and loss of Vamp1. So to backtrack a little bit, I'm sure you all know, but I'll remind you. So the Vamps are also known as synaptobrevin, which I'm sure you're very familiar with. It's part of the SNARE complex, which is found at the synapse and functions to bring synaptic vesicles to the cell membrane and release their contents in order to release a neurotransmitter. The Vamp or synaptobrevin that most of you are probably familiar with is Vamp2, which is generally also referred to as synaptobrevin in many cases and is found in all neurons. However, the target that we're interested in, Vamp1, is a little bit less studied, but it has a high sequence similarity to Vamp2 and functions in a similar way. So since not so much was known about Vamp1, we were interested in just understanding where is it expressed and what is its expression pattern. So if you just look at neuronal development across from embryonic stages to adult, we find that Vamp1 is expressed a little bit later than Vamp2. In fact, it doesn't come up really until 2 weeks postnatally. So that was a little bit different. And then we were also interested in the immunofluorescence pattern of Vamp1 compared to Vamp2. So to do this, we use wild-type cultured hippocampal neurons. And when we stain for the two Vamps, we find that while all neurons express Vamp2 in discrete puncta, as has been shown previously, many cells don't express Vamp1. However, a small subset of cells, about 10 to 15%, express both Vamp1 and Vamp2. So this made us think that perhaps Vamp1 is specific to a particular subset of neurons, such as the GABAergic inhibitory neurons. And so to test this, we again did immunofluorescence in wild-type cultured neurons, looking at colocalization of Vamp1 with markers of inhibitory markers or excitatory markers. So here I'm showing you our colocalization of Vamp1 with the inhibitory markers GAD65 and 67, and you can see that they colocalize very well. And that is shown here. However, when we look at colocalization of Vamp1 with the excitatory markers v-GLUT1 and 2, we find that they have virtually no overlap. And so from this data, we could say that Vamp1 seems to be specifically expressed in GABAergic inhibitory neurons. And we could confirm that this is also true in vivo. So here I'm showing you sections of adult mouse hippocampus from wild-type and Rbfoxl knockouts. Vamp1 is shown here in red in the pyramidal layer of CA1. And it, again, colocalizes very well with the inhibitory marker GAD67. As expected with loss of Vamp1, with reduction of Vamp1 in the Rbfoxl knockout, we get a significant decrease in the overlap of Vamp1 with GAD67 in the knockout due to loss of Vamp1 positive puncta. So we wanted to make sure that the loss of Vamp1 was due to a general reduction in Vamp1 expression and not necessarily in loss of excitatory or inhibitory synapse numbers. So to test this, we went back to cultured hippocampal neurons in wild-type and Rbfoxl knockouts. And in those two conditions, we assayed excitatory and inhibitory synapse density by colocalization of synaptophysin and PSD95 for excitatory synapses, and GAD65, 67, and geffen for inhibitory synapses. And in both cases, we find that loss of Rbfoxl does not have any significant effect on the number, on synapse density. So then, of course, the interesting question is what electrophysiological changes might we see with loss of Rbfoxl? So in collaboration with Weizheng Wei and Tao Model, we performed basic electrophysiological assays on acute hippocampal slices from the wild type and the Rbfoxl knockout. We first tested miniature inhibitory postsynaptic currents in CA1 of wild type and Rbfoxl knockouts. And we find that while there is no significant change in amplitude, there is a significant reduction in mini-iPSC frequency with loss of Rbfoxl. So this made a lot of sense in the context of knowing that Vamp1 is a SNARE protein and has been shown to regulate synaptic transmission, and that Vamp1 is specifically expressed in the inhibitory neurons. However, we also tested mini-iPSCs, and we were surprised to find that there is also a significant reduction in the frequency of mini-iPSCs in addition to that of the mini-iPSCs. And we had not expected this from what we knew of Vamp1. So to reconcile this data with what we knew of the Rbfoxl knockout, which is that it has increased seizure susceptibility, we decided to look at the excitation inhibition ratio between wild type and Rbfoxl knockouts. So in this experiment, we are looking at the evoked response of the excitatory and the inhibitory component, and we take the amplitude of each component to get an EI ratio. So when we compare wild type and Rbfoxl knockouts, we find that the EI ratio is still skewed towards increased excitability, despite our previous findings that both mini-iPSC and iPSC frequencies are reduced. So then we were particularly interested in understanding the function of Vamp1 and to confirm that loss of Vamp1 could affect inhibitory synaptic transmission. So to do this, we went back to our cultured neurons and we used AAV to express shRNAs to knock down Vamp1. And then to test the role of Vamp1 specifically in inhibitory neurons, we then used this mouse DLX enhancer, which drives expression specifically in GABAergic neurons, to specifically re-express Vamp1 only in the GABAergic neurons. So the Western blot for that is shown here. And then this is just showing you the experimental setup. So we have a control shRNA. We have an shRNA against Vamp1. And then in our rescue experiment, we will re-express Vamp1 only in the inhibitory neurons. So what we find from recording, again, mini-iPSCs and iPSCs in these cultures is that while amplitude is not affected at all in any of these conditions, loss of Vamp1 leads to a significant reduction in mini-iPSC frequency. So that's great. And we can rescue the change in frequency by adding back Vamp1 specifically in the inhibitory neurons. Surprisingly, when we look at excitation, looking at mini-iPSCs, again, amplitude is not affected, but similar to what we saw in the Rbfoxl acute hippocampal slices, knockdown of Vamp1 also leads to a significant reduction of mini-iPSC frequency, and this reduction can be rescued back to wild-type levels by adding back Vamp1, but only in the inhibitory neurons. So this was particularly interesting because it meant that perhaps this was a homeostatic effect where losing Vamp1 and therefore losing a lot of inhibition, the cells try to compensate by reducing their excitation. So, of course, since we had seen changes in inhibitory synaptic transmission in the Rbfoxl knockout, and then we can see in cultures with Vamp1 knockdown that Vamp1 can also affect inhibitory synaptic transmission, we were very interested in knowing whether or not we could rescue these defects in the Rbfoxl knockout by adding back Vamp1. So to do that, we come back to, again, our AAVs expressing Vamp1 under the control of the DLX enhancer, which will drive expression specifically in the GABAergic interneurons. We used, as a control, our wild-type littermates with one side left uninjected, the other side with a control injection. And then in our the samples that we're interested in the Rbfoxl conditional knockout, we have a control side injected, and then the other side injected with the inhibitory neuron-specific Vamp1. And this is just showing you that the DLX enhancer in vivo is also specific to GABAergic neurons. So, again, with the help of Wei Zhang and Tom, we made acute hippocampal slices from these wild-type Rbfoxl knockout and Vamp1 rescued animals. And looking at mini iPSCs in CA1, we find again that inhibitory amplitude is not affected. However, with loss of Rbfoxl, we get again a significant decrease in mini iPSC frequency, and this change can be rescued back to wild-type levels by adding back Vamp1 specifically in the inhibitory neurons. And most surprisingly, we found that we could rescue EPSC frequency similar to our culture experiments by adding back Vamp1 specifically in the inhibitory neurons in the context of the Rbfoxl knockout. So this really confirmed to us that in our first ePhys experiments, where we saw a decrease in mini EPSC frequency, that was likely due to a homeostatic change as a response to loss of inhibition. So to summarize for you the second part, we found that Vamp1 is specifically expressed in inhibitory neurons, and that loss of both Rbfoxl and therefore a decrease in Vamp1 expression results in reduced inhibitory synaptic transmission. And that the EI balance in the Rbfoxl knockout hippocampus is skewed towards increased excitation. And finally, we show that increasing Vamp1 levels in vivo in the Rbfoxl knockout can rescue the defect in inhibition. So together with our mechanistic data, what we think is happening is that in your wild-type brain, in the wild-type inhibitory neurons, you have a good level of Vamp1 expression due to Rbfoxl binding in the Vamp1 3' UTR, promoting its expression in part by blocking the repressive activity of miR9, and that will give you a good excitation-inhibition balance. When you have loss of Rbfoxl, you have a decrease in Vamp1 levels, also in part by the not having blockage of miR9 activity. And that reduces inhibition in the brain, skewing your EI ratio towards increased excitation. And we show that if you can somehow increase the levels of Vamp1, specifically in the interneurons, you can rescue this EI level and inhibitory synaptic transmission back to the levels of the wild type. And so with that, I'll give my acknowledgements. So first, thank you for coming and listening to my graduate project today. I have to thank Doug for being a really wonderful graduate mentor. The lab present and past for making the lab feel like my second home. Beverly in our lab did all of the bioinformatics. Andre and Jeanne and Kelsey Martin's lab performed the iClip analysis. Reem helped me with experiments for the project. Our really wonderful collaborators, Tom Odell and Wei Zhang Wei, who did all the electrophysiology that you saw here today. Bal's lab for teaching me how to do injections, and of course, collaboration from Rbfoxl for taking part of our journey with us through the review process. And of course, our committee at UCLA and our funding sources. So thank you for listening, and I'll take any questions.
Thank you for a lovely talk, and I open to, yes.
So when we increase the level of Vamp, why doesn't Rbfox bind? Because Neuron is not binding anymore, right?
So your question is, if when I re-express Vamp1 in the inhibitory neurons, why doesn't
Rbfox bind to it now?
So the construct that I put back in, maybe this is better. The construct that we put back into the Rbfoxl knockout does not have a 3' UTR, so it's the coding sequence only. So that expression is independent of both Rbfoxl and Mir9. Does that make sense?
Yeah.
Did you look at some of the other SNARE complex proteins and see whether their levels are altered or not?
Yeah, so from the list of gene expression changes that we had, I think in terms of SNARE proteins, Vamp1 was really the only one that was called as significantly changed. But in general, there are a lot of other synaptic proteins whose expression levels are changed with loss of Rbfoxl1, which very likely contribute to the phenotype. But I don't remember significant changes in any of the other SNARE proteins.
Did I catch it correctly that Rbfoxl1 does not regulate Vamp1? It's your prior brain region that you studied at Brown with cerebellum?
Ah, yeah. Yeah, so it doesn't. Well, okay, so with loss of Rbfoxl1, we don't see a decrease in Vamp1 in the cerebellum. And that is a really interesting question that we haven't really gotten to. There are a multitude of possibilities why it could be happening. It could be that there's different levels of Mir9 in the cerebellar inhibitory neurons, or it could be that actually you have differing levels of the Rbfoxes, Rbfox1, 2, and 3 in the cerebellum, and it's not controlled by Rbfox1, maybe it's controlled by Rbfox2. But it's an interesting question that we want to look at, but we just haven't gotten to. So.
Can you use the model of homeostatic plasticity with cultured neurons to see if under certain conditions Vamp1 levels change, and if that has anything to do with the interaction between the Vamp1 RNA and your Rbfoxl1?
Yeah, so that's a great question as well. So we haven't really seen anything significant with Vamp1 regulation in homeostatic plasticity in culture. We are, as a sort of a side thing, we are interested in stimuli that would change the ratio of nuclear to cytoplasmic Rbfoxl1, which might be another way to regulate Rbfoxl1 targets under activity, changes in activity. So I guess my answer is, for homeostatic plasticity in cultured neurons, we haven't seen a change in Vamp1 driven by Rbfoxl1. It might be different in culture than it might be in vivo. So it's something that we're interested in, but we haven't seen it yet. So.
Have you looked at the regulation of different classes of inhibitory neurons?
Yeah, so that's another interesting question. I think Rbfoxl is more interested in that and probably is better suited to answer that. They're looking at PV interneurons and somatostatin Rbfoxl regulation in those two subclasses. We haven't looked at that, although I think we're possibly interested in looking at a broader difference, excitatory versus inhibitory neurons and Rbfoxl regulation in the cytoplasm between those two classes. But yeah, if we could do that, that would be great, because I think what we've shown here is that between the broad classes of excitatory and inhibitory, there is a difference in Rbfoxl targets and regulation, and it's likely that as you go further in the subclasses, there's also differences in Rbfoxl regulation.
You do see Vamp1 in the other classes.
So from my staining, I can say for sure that Vamp1 is in the PV interneurons. From looking at the anatomy, it's likely that Vamp1 is also in SST neurons, but we haven't looked at more specific markers to find out if it's in even in more particular subclasses.
So I should remember that from your talk that both Vamp1 and 2 are in inhibitory neurons?
Yes.
So what do we know biochemically about differences between Vamp1 and Vamp2 and how they regulate secretion?
Yeah, so there's not much known about Vamp1. Basically, I think the best published data for this looks at the neuromuscular junction. So motor neurons express both Vamp1 and 2, similarly to inhibitory neurons. So at the NMJ, you need both 1 and 2 to get reliable release. However, the motor neurons can still release neurotransmitter with loss of Vamp1. It's just more variable.
So is it thought that they're basically simply functionally equivalent paralogs, or is there something that they are regulated in different ways? Do they have some different domains or phosphorylation sites that look interesting?
Yeah, so we haven't looked at that. They're at the protein level, they are quite similar. I mean, there are differences, but we haven't looked at phosphorylation sites or anything like that. But basically, it seems from previous experiments that motor neurons need both Vamp1 and 2 to get reliable release. And I think...
That could just simply be that they're paralogs.
Yeah.
But you think they will have some interesting differences in how they function?
Yeah, because, so for example, inhibitory neurons, the question I've always had is why do you need both? And so if you look at PV interneurons, they need very reliable release, because it has to be fast spiking many times. And so that might be one reason to have both Vamp1 and 2. But in terms of the kinetics of Vamp1 versus Vamp2, I don't think anyone has quite looked at that, but that's an interesting question.
So we don't have any other questions, and I'm sure there are plenty more that we can ask them outside. Thank you again for a wonderful talk.
Okay, thank you.